Clostridium, didn't we? I think. This one?
I think yes.
That's what I think. So I'm going to tell you in here, you know, I'm putting different genera in here that may show up, but I will tell you that I'm emphasizing certain species. And it might be like, oh, I've heard of that one. Great, good. So, you know, add a few extra notes to that. That's fine. What I'm also hoping to doing, though, is by presenting a phylogenetic approach that you kind of see some of the connection, the same way that when you learn plants, oh, you know, this is an angiosperm and it, you know, does this, and this thing. So it adds a little bit to that. Okay. In some cases, though, I understand in microbiology, it can be kind of confusing because you get, like we were looking at with the gamma proteobacteria. You know, why is E. coli and this other thing in the same group? Sometimes that's the deal also, but that's the state of it. I mean, if I could find a better way of explaining it, I certainly would. It's the deal on here. So let's go with what we have in this. And what we want to start talking about, now we're in that quiet...
Oh my gosh.
Sorry. Hey, anybody going to St. Louis next Friday? Well, if you want to come to St. Louis on December 2nd, we're having our show at the Science Center. So if you want to come by, Jack and Mike Miller and a few of us, we're doing that photo stacking stuff. But anyway, if you want to come down, we'll be doing it. That's what this call's about.
I hear you.
Huh? Yeah, well, the connection, yeah. There's a lot of pressure now to actually think about what we're doing, which I've been thinking about it for 3 months. Okay. Phylum Firmicutes. Were you thinking, what was the connection? Is that what you're wondering?
Actually, she said something.
Yeah, he's free-forming again. Okay, Spora Sarcinia. The only reason I have this genus in here is to remind you that we keep seeing this root word, and now we know what a Sarcinia is. I'm not going to emphasize that one. We're going to emphasize the ones that you probably know something about, Bacillus and Clostridium. Okay, they do have very, very distinctive shapes. Okay, of all the rods in the bacterial world, Bacillus gets named for the rods because they were common. You know, they were finding these. You know, they're starting to look at stuff. You know, way back. You know, in you know what occurred in soil, what occurred in pond water and stuff. Bacillus came up a lot. Okay, you may know Bacillus as something that has a lot of exoenzymes, probably the ones that we used in the lab. It degraded more different things, right? Starch. We talked about degrading cellulose. It'll degrade protein. You know why? It lives in the soil, and it needs to like be chewing on stuff, and it doesn't always have the right stuff to chew on. Like E. coli can pretty much wait for you in your next whatever, your next slice of bread or whatever to ferment, and if it's in your body, it's doing really well. So Bacillus Clostridium is one of our favorite anaerobes. Okay, both of these are spore formers. So, and their spores are endospores. Okay, we'll be talking, if we haven't already, about myxospores and a few other kinds of spores. So endospores, just to recall for you, is inside. Now the position of the spore can tell us a little bit about the bacteria. Why do bacteria make spores? Why don't they just make more bacteria, which we call the vegetative form of the bacteria?
Because in case there's like a harsh condition where they might not be able to get nutrients or grow for a while.
Yeah, it's their little time capsule, or whatever you want to think about it. I mean, it is their, you know, the future. It's a very resistant situation. These are not motile, so when things go bad, it's not like they swim away or attach or do something. You know, they produce a spore, and that is very hard to degrade, and therefore hard to sterilize. We talked about how pasture kind of got messed up with spores for a while. So, good. So spores versus vegetative cells. And here's some pictures of them. The little needle thing, some people think of it as a spoon, generally depicts a terminal spore, in this case, as opposed to a central spore. So this is Bacillus, and these are different types of Clostridia. And the reason what you're seeing here it's still light microscopy, it's not an EM. But what you're seeing is a general stain. I don't know, it's a black and white picture. Maybe they use methylene blue, maybe they just use crystal violet. The point of it is, and if you're in lab, to get stain into those spores, you know, what we had to steam it in there, and we had to use a real small kind of stain size called malachite green. So the thing is, we tend to see these clear areas just because they don't stain, and the light, this transmission, light comes through. So they're very characteristic that way. Okay, mainly bacillus here. Some of these other names are in here. Take them with a grain of salt. Already talked about this. Very easy to find bacillus, and they do a great job of breaking stuff down. Not hard to find at all. Some bacilli will produce antibiotics, but we're going to associate antibiotics mainly with streptomyces, which is coming up maybe later today. We'll see how far we get. I want to talk about Bacillus thuringiensis with you. We talked about the spore, just a reminder on that. So we'll come back to Bacillus thuringiensis. Clostridium, you give Clostridium oxygen, it stops. It's an obligate anaerobe. Okay, don't get that mixed up. Oxygen doesn't make it explode or die, but it inactivates itself. It just won't work. So mainly found anaerobic pockets in the soil. Also lives in the mammalian intestinal tract. Yeah, you have some Clostridia in you. Usually not a real big problem. But what's the big problem now? What species of Clostridium is showing up in hospitals and with people? Probably a lot more. I didn't remember hearing it as being a bigger problem as now. Hmm? Well, botulism is it showing up more? Yeah. Sometimes it'll show up like SIDS. It'll kill kids. Have you heard of C. diff? Maybe you haven't heard of Clostridium difficile. C. diff is kind of like, we abbreviate everything now. We can't say people's names. We say, oh yeah, that's number 54. Anyway, the C. diff is Clostridium difficile, and that's showing up a lot. Do you know why? What are the reasons why it shows up in people? People start taking general antibiotics, or trying to knock out some kind of thing in people's bodies, and a whole bunch of your natural flora gets knocked out. And it turns out C. diff can then be an opportunist and take over the intestine, and it causes diarrhea, terribly foul. I mean, what comes out that end is not a rose anyway. We all know that, but mainly really bad smell and so forth. So C. diff is on seems to be, those occurrences are on the rise, mainly due to antibiotics on that. So we talk about a few different diseases of Clostridium. Let's start talking about different species. How many of you know about Bacillus thuringiensis? In Illinois, this is like one of your big GMOs. What do you know about it?
I don't know much about it.
Don't know much. I just heard about it. Okay. Anyone ever hear in Illinois about BT corn? Have you heard about that? Okay, that's Bacillus thuringiensis. Okay, it's not that the corn is bacteria. That's not it. But they took the gene out that will be an insecticide. Okay, and they abbreviate BT corn. It's a GMO. We live in one of the most massively industrial areas of the United States. Probably surrounded by more genetically modified organisms than probably anywhere else, or at least we're tied for some places. Okay, so how that makes you feel is up to you, but be aware of it. The industry is massive and needs constant looking at. So with BT corn, when they did this, they actually just did shotgun cloning of that gene. They basically blasted it into a bunch of different corn until it took and expressed. So it wasn't done in a highly controlled level as I remember it. The last geneticist here, Roy Mosher, explained it to me, and that was in 1999 when we were talking about it. it's a fun thing to do. You can get corn, you guys now know enough about genetics, you can get online, find the 16S for the BT, make yourself some primers, and go in, extract DNA out of corn or whatever you want, and see which taco chips have BT, which ones don't. A few years ago, Taco Bell got in a big lawsuit because they, BT corn showed up in their taco shells, and they said, no, we don't buy, at the time, we don't buy BT corn. How can this be? And then somebody said, you know, the pollen blows around, so, you know, one of those things where, well, it's not the seed we put in there, well, nature does. So what is the deal with Bacillus thuringiensis? It produces a protein crystal. Let me get some words up here for you. So it produces a crystalline protein, okay? So now, if the bacteria isn't there, the gene produces this little protein, it's a crystalline. And when the bug is out there, you know, on a hot summer night, just wants something to eat, it's a little thirsty, all it wants to do is have a nice bug life. It comes up and it eats the corn plant, just sitting there nibbling on it, and then the BT protein goes in its gut and it makes little bitty slits, okay? So then through the night, the bug desiccates and dies a horrible, painful death, alone in a farm field all by itself. That's it, that's BT protein. So, anyway, whether you believe that or not, you'll probably never forget the mechanism of the moth, but that is right, they die from desiccation, okay. Tends to be, you say, well, why don't we just do that for all the bugs? I looked into that. It seems that if the gut is more alkaline, that it tends to, or neutral, it tends to be more effective. And then people start saying, well, what about that BT corn? You know, those people are, the farmers this time of year are getting all the corn and they're chopping up all the stuff in the back. Won't that affect all the insects that eat that stuff? And Matt Wiles down at SIU said, yeah. He and Jennifer Tank at Notre Dame said, we're going to look at that. And because the idea is that the protein breaks down right after you chop up the corn and stuff, which was only partially true, as most propaganda is. So the thing is they found out that it will affect the caddis flies and the mayflies and things that might get the BT corn in streams. So not to the effect where it's wiping stuff out that you never know, like the eagle and DDT, but it seems to have an effect. So, but if you want to look up that, Wiles is a really nice guy. Jen Tank, of course, is famous for all her work on nitrogen. If you're looking for someone to talk there, really nice people if you ever have a chance to meet them. So that's the BT corn story coming from Bacillus thuringiensis, one of the more famous of the Bacillus crew. Okay, let's talk about a few famous Clostridia. I'm completely off on my time now. Oh, we don't have much time together at all. Well, let's talk about this. We talk about Clostridium, where to find it. The big thing with Clostridium is that it's interesting. It is an anaerobe, so where you probably grow a lot of Clostridium is in the base of the Winograsky column. They do really well metabolizing the anaerobic decomposition of cellulose. Aerobic is pretty easy. You know, we get bacteria that bite off pieces of the end, by hydrolytic enzymes that clip off the ends. Some go on the inside, and they start breaking cellulose down. Okay, if you're a termite, you hang on to those bacteria and some of those protists in your belly so that they'll break down that beta glucose bond. You don't talk about as much in any parts of ecology, but the anaerobic decomposition of cellulose produces a whole bunch of fatty acids and carbon 1 chains. And these really are a whole nother way of a breakdown. We don't tend to look at it in nature because it doesn't happen as fast. Aerobic is still aerobic. It's great. You know all about the energetics of that, why it goes faster. But anaerobic produces so many other breakdown products that it should be looked at a little bit more. So, anaerobic rods, spore form. Botulism. Now, we were just talking about that. So let's some here. The spores in the honey is a thing. I didn't know that was starting to reoccur. It was one of those things that I thought people had figured out and stopped doing it. A lot of times you'll see in your honey it's pasteurized. That might not knock out spores. Really, you have to look at how people work with it. The other thing is that anything you give to kids, their immune system, even up to like 5 years, kids, they're constantly, they get sick, they get a fever, they get better for most things because they're building that antigen antibody memory system in their body, and they have youth going for them, so it's really cool. But you get caught with a big explosion of botulism in anything, but with kids, you can knock them out. So it was related to SIDS, and I did not know it was on the uprise. That is sad. And do you know why it is on the uprise? Is it the source, or okay? I don't know. That'd be a good thing to find out. I kind of hope that was behind us. Kids dying from botulism. So what is the main way that botulism works? Most times it's ingested with food. Clostridium is in dirt. It's in soil. So it's easy to have it with rooted vegetables. If you're canning and you're not, and you're canning something like tomatoes, they're very acidic. You don't have to worry about doing a lot of boiling and stuff, but pressure cooking other foods, whether it's at Del Monte or Green Giant or in your kitchen, you're trying to kill spores, is what you're doing in the canning process. What happens here is the spores will germinate, and it affects the neuromuscular synapse. Basically, it blocks the release of acetylcholine. Okay, so your muscles stop working. So what will happen? You'll get slurred speech in the beginning. Droopy eyes will be some of the first symptoms. And then botulism will basically, you stop working. You become lethargic. But I don't know why it does not go after your heart and the heart synapses. Do you know?
It does. Like, what it does is that it targets first, like, you'll get symptoms of, like, fever, vomiting, nausea, diarrhea, and then you'll have numbness and tingling in the face and then extremities, and then it'll slowly start going to your lungs, so the diaphragm stops.
The diaphragm is the part that I thought you died from. I didn't think that you died from your heart stopping.
I don't think it really does target the heart. I think you just you
Right.
have to be on, like, a ventilator, and you have to keep your heart can stop because you're not dying.
Right, but I mean, of the synapses, do you know some of the synapses, you'd think the heart would be like, okay, no acetylcholine, that'd be the one.
The only thing I think of is that the heart has a different way of.
It's electronic, right?
Yeah. It has like the Purkinje fibers, so it doesn't always.
That's what we thought about in the nursing class, and I never had a chance to look it up. I think now that you mentioned that's right. So I do know that the diaphragm is the thing that kills you. You tend to suffocate from botulism. Now, if you get a good amount of botulism, you'll be going like, oh my God, I have, it's going to go pretty fast. But in milder cases, as you say, you can work through it, get to a hospital, and have different other things help you make it through the poisoning. Foodborne botulism basically was related to canning. I don't know that a lot of people can anymore. Maria cans everything all the time, okay, and that's why I never eat anything, because I don't want to get botulism. No, it's all good stuff. But what would happen is that with canning, especially with proteinaceous material, is that the spores would get into the canning. If it wasn't pressure cooked and completely cooked, the spores would germinate, and then when you ate the, whatever, in Alaska it was a lot of canned salmon is what we did up there. Also, meats would be the big thing. I can tell you a story about the Depression. My grandmother would tell us that we call everything tin cans now. There's very little tin in a tin can. It's too expensive. They use that for your computers and stuff. But back then, cans were coated in tin so they wouldn't corrode. They were still metal. And if a can became dented, they would either, it was the Depression, they still sold it, but they sold it at a discount. Okay, so people potentially could have spores between the tin and the metal that would germinate, but they would take the chance because they were also starving to death. They would get the stuff on sale. The reason I know this is as a little boy, I watched my grandmother actually dent cans to get a nickel off of them. Not that she's dishonest, it was just, she'd been through the Depression, and I think stuff like that just really messes with your mind. You know, just you waste nothing, you save everything kind of thing. But sometimes, yeah, there's other good stories about moonshine and stuff in that era, but the botulism one comes to mind now. So, yeah, if you have low acid and so forth, you don't have to worry about that as much. And we've talked about that. Around here, where you come up with botulism, and maybe some of you people who go outdoor more know about this, but around the rivers, we have these floodplain lakes, and if you've been around, these lakes are very shallow and large, and they'll connect with the river. A bunch of fish will go in there, carp, whatever, and then the lake will go down, and they get trapped, and then it goes down more, and you have a bunch of die-offs, either from no oxygen or just they run out of water. Well, now you have a proteinaceous source in these anaerobic muds, and then you start to have sprouts grow up. So every year we get a lot of avian botulism. Anyone who follows like ducks and duck biology, it shows up a lot, and it's a very it's actually something that, you know, if you want to look into that. I know the Fish and Wildlife have asked me several times to look in, and I just haven't had the time, but people are very concerned about that kind of process, activating botulism and just killing off a bunch of ducks. Kind of hard to control the Illinois River. You'd say, oh, just don't let that happen. Well, yeah. Okay. The opposite, the relaxation, is to have the muscle fire and never relax. And that's the cousin, I guess, of botulism, is Clostridium tetani. Okay, same kind of things, germination, wherever you're going to get it from. You know, I don't know what you. When I grew up, my mom would say, I love my mom. But like, okay, you kids are playing outside today. Be careful. Don't step on any rusty nails. As though this was the I think about now. This is the neighborhood I grew up on, where you could step on rusty stuff, and your mom was concerned about it, which is kind of funny. Like now it's like, don't forget to wear your helmet. It's like, don't get shot today. Okay. So anyway, did you guys ever? Did anyone ever tell you about like a rusty puncture? Okay, and tetanus. Yeah, and that's the deal.
I actually had that happen to me.
You did, and so the thing is, and my question is, why were people worried about a rusty object and tetanus? It's a botulism thing. The thing is rust by itself isn't going to hurt you. It's ferric oxide. It's rust. You know, you eat it in your vitamins in some kind of form or the other, but that's a different passage through your body. The thing is that where nails rust is usually around soil, you know, and then you inject it straight in your bloodstream. And all it takes is a few spores, and all of a sudden, you know, in severe cases, you end up like this poor soul. What happened to you?
I stepped on a rusty nail.
And did you get
I don't remember if I got tetanus, but there was like a pustule on my foot, and I had to get it like popped at the doctor.
Did you get it? But you got a tetanus shot like right away, right?
I think so.
Yeah. If you don't have your tetanus updated, they'll nail you right away. I mean, you got nailed. Yeah, so that's the deal, because, it's like, what's it, why do nails, and years later, Mom, why do nails have tetanus on them? You start to kind of put it together after you get your PhD and are sitting alone figuring stuff out. So lockjaw, as you can tell, that was really a problem when people, again, in warfare, people aren't necessarily clean with their spears and stuff, so people would get nailed with that. Same kind of thing, that, now it tightens up your diaphragm, the throat and so forth. Suffocation usually takes you out before anything else, yes.
So what if you get cut by metal that hasn't been exposed to soil? Would you still need to get your tetanus shot?
Chances are no. You accidentally touch yourself with a razor blade. Just making this up because that could happen. Razor blades are fairly common and not often regulated enough. Chances are no, that you wouldn't get tetanus. Okay.
I work in a factory. I sort metal.
So what's the other part of the story?
I sort metal parts and I cut myself the other day.
Yeah, chances are, you have a cut. Chances are you could have an infection from something else. Chances of getting tetanus, low. Okay, unless the parts are sitting in the yard in soil first, and then they come in and cut you. More was true if you probably work in like an auto place or something, or salvaging things.
I know that anybody that comes into the ER, if they have any form of cut,
Yeah.
and they're like, are you immunization up to date? When's their last
Yep.
shot? And then they're like, I don't remember. Yeah.
Yep, absolutely. That's the deal. We did that because it's so easy to get the shot and stop the toxin, as opposed to trying to treat it once the toxin is expressed. That's the deal. I mean, I was going to say like rabies, but you don't take any chances of rabies. You just die. So, okay. Here's another one, Clostridium perfringens. I'm sorry. 940, okay. I did something to my wrist. Does anyone know about broken wrists? You probably know about broken things. I need someone to look at my wrist, but I don't want to go to the doctor. Anyway, I took my watch off, is the point. That's the point. I know I'm free-forming today, I'm tired. Okay, so gas gangrene comes from Clostridium perfringens. Okay, these have hemolycins. What are those? And maybe it's up there, but do you remember? It's in the name, lysis of blood. There you go. Okay, lysis of blood. And a lot of enzymes. The deal is we know at best gangrene infecting flesh. Okay, sometimes frostbite victims will later get gas gangrene, usually with an abrasion or something that introduces it. Probably some of the most famous cases, unfortunately, or infamous cases, would be like the Civil War, where we had at the time, modern armaments with really kind of like two generation old strategies of using them. So not only was it bad enough that you were getting hit by this huge, relatively slow-moving metal lead object, that may or may not have killed you. If you live, you get these very bad wounds. Okay, and what happens is most wars, but not terribly clean. Had Listerine wasn't over here yet. Okay, people weren't thinking a lot about sterility and even washing their hands before surgery sometimes. So what you would have is the infection by Clostridium perfringens. And you go, well, yeah, but it's in a muscle. There's vascularization. Well, scabbing, right? Now you have an anaerobic situation with the expression of the spores. You have not only then the infection into the wound, you have extracellular enzymes and hemolycins that are making the wound greater. And these guys produce a lot of gas. I don't know if it's up here, but gas, gangrene, yeah. So what they'll do is if it doesn't expand the wound, it would start to explode it open, thus making the wound even worse and definitely more painful. And one of the reasons there was so much amputation was because of that, is to try to cut away and above it before it got infected was the deal. That is still an option. you know, but if you get gangrene soon enough, depends upon, you can intend to treat it. So, yes, you could get it by eating stuff. People are zipping things. I think you're trying to have to go soon. Oh, I just found, this is kind of an this is just, one, you gotta go. Okay, just a few things. This is kind of interesting. We always talk about, are there any gram-negative spore formers? No. Are there any spheres that makes ones? And here's a Sarcina that makes spores. That was the big thing here. So it's kind of interesting to see. That's my big thing on that slide. And we go into mycoplasma. I guess we'll pick that up a week from today. We only have 2 lectures left, okay? I'll try to get some review stuff up. Try to get all your stuff corrected. That's on my plate. So come to lab. I think we'll have a little fun. I don't think it's going to take us 3 hours, but we'll do some things. We'll look at last week's stuff. We'll do a little exploration of eukaryotic organisms. Not to death, but we'll do a little exploration, have some fun, and call our quits for the...
Do we have open lab?
Huh?
Do we have open lab?
Yeah, open lab. Is it Tuesday? Yeah, starting at 10.
Okay.
Soon.